Hello, everyone, and welcome to today's webinar. I'm Nathan Nee, Senior Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speakers Perla Vega and Philip Kitchen will discuss how to use mass photometry in protein purification workflows, sample characterization prior to cryo-electron microscopy analysis, and G-protein coupled receptor solubilization. And with that, let me introduce our first speaker. Perla Vega is a sales and application specialist at Refeyn. Vega completed her PhD in biosciences at the University of Birmingham, focusing on the characterization of myobacterial cell wall components using a variety of bioanalytical tools and microscopy. Vega then moved into industry to develop assays for single molecule microscopy before moving to her current role at Refeyn, where she built strong relationships with industry and academic partners to accelerate and enable biomolecular characterization. Perla, the floor is yours.
Hello everyone, I'm Perla Vega, Technical Sales Specialist for Refeyn, and today I'm going to be showing some data on how mass photometry can help to rapidly characterize complex samples. At Refeyn, we are the company that commercializes and develops a technology called mass photometry, which is an innovative biophysical analysis tool that measures the mass of single particles in solution, without the need of any label or immobilization procedure, and within minutes. The ability to measure the mass of single molecules in a bulk sample means that mass photometry can provide access to subpopulations, which makes it possible to determine complex stoichiometry and oligomeric states. It also means that it's possible to study interactions, and even complex multi-step processes are accessible for study with mass photometry. Furthermore, the single molecule counting over a wide mass range and high dynamic range enables the detection of low abundance species and the characterization and monitoring of sample heterogeneity. In this slide, I would like to show you the mass photometry mass range. So the smallest particle that mass photometry can detect is a protein of 30 kilodalton, but then it can go all the way up to 5 megadalton plus. That's why mass photometry can be used to measure any particle within this mass range. In fact, all these type of biomolecules or particles have been characterized using mass photometry. In terms of how mass photometry works, well, this can be explained with the follow diagram. This gray line represents a glass surface, and on top of that glass surface, we put a mix of particles that we want to characterize. The diagram is showing, let's say, protein or protein complexes of different masses. What's going to happen in our system is that we're going to shine a laser through the sample, and some of that laser is going to be reflected back to the objective by the glass cover slip. And as you probably know, every particle that interacts with light scatters light. What is going to be detected in a mass photometer is the interference between the scattered light by a single particle in the glass-water interface and the reflected light by the glass cover slip. That interference is going to be detected as a change in the contrast, or this dark spot that we're seeing here. We like to call it mass photometry contrast. And the interesting bit of mass photometry is that the intensity of this contrast, of this change in the contrast, scales linearly with the mass of the particle. So in a typical mass photometry experiment, what's going to happen is that we are going to detect the signal coming from particles landing into the glass cover slip for a period of 1 minute, quantify their contrast or measure their contrast, and then transform that contrast into mass to ultimately provide a histogram that shows the mass distribution of the single particles that we have in the sample. Overall, operating a mass photometer is very straightforward, and the data that mass photometry produces is also very easy to interpret. Here, I'm going to show you how typical mass photometry measurements look like. So we start seeing the lid of the instrument, then we see the objective. On top of the objective, we see the glass or the sample carrier slide onto which the user is going to pipette some microliters of the sample. And that's going to happen now. Okay, now it's going to give it a quick mix. And as soon as the user closes the lid, which is going to happen now, where the system is going to start detecting the signals coming from single particles binding into the glass cover slip. So the recording normally takes 1 minute, and after 1 minute, the analysis is pretty much done. Overall, doing a mass photometry experiment is quite straightforward. Because mass photometry measures mass, this makes it a very versatile technology, as mass is a universal property. This versatility is a major strength because mass photometry can provide useful information on many different types of samples, so it can be used in many different ways. We can focus on the composition of the sample as a whole, where we want to assess aggregation, filling ratios, or sample purity. We can also use mass photometry to gain insights on how our samples are forming complexes or oligomeric states. And we can also use mass photometry to understand how our species interact with each other. In the next set of slides, I'm going to be looking at selected subset of applications that I put together, as I believe that this will be the most relevant for this audience. But please be aware that there are plenty other applications that are not going to be covered in this seminar. Let's start on sample characterization, where the aim is to understand the sample as a whole. First, I would like to show some data on how mass photometry can be used to monitor a purification process. This data was generated by Microshalom Lab at the WIS, and it shows how mass photometry can be used to monitor the purification of the 20th subunit of the rat proteasome. This particular protein complex is rather large. It has a mass of 700 kilodalton, and in these histograms on the left, we can see how mass photometry was used to monitor the relative abundance of the 700 kilodalton mass range across different purification processes. If we were to plot the molar fraction or the percentage of the species that we wanted to characterize, meaning the 700 kilodalton species, we can clearly see that after the fourth purification step that corresponds to this buffer exchange, it's where the highest yield of the species that researchers wanted to purify is showing. So there's clearly not a need for a fifth purification step, and the purification protocol can be shortened. This shows how mass photometry can help you to understand or monitor better your purification processes using minimal sample condition. During antibody development, aggregation is a critical quality attribute that needs to be monitored. In fact, we generated this information with RIC Biologics, and here we are showing how mass photometry can be used to quantify antibody aggregation for different clones during the development of biosimilars. So for this slide, the supernatants containing the antibodies were purified using a protein A affinity column and measured by both CQV and mass photometry. In the bottom, we can see a couple of tables that show the percentages of monomer dimers as measured by both CQV and mass photometry. And in fact, we can observe that the percentages of the monomer and the dimer as measured by CQV and mass photometry correlate very well, or are very similar. In fact, both techniques show that clone 3 and clone 10 have the largest dimer fraction. But it's definitely worth highlighting that these mass photometry measurements required only 30 nanograms and took around 1 minute, whereas the CQV measurements were done with 10 micrograms and took around 30 minutes. In summary, mass photometry generates information on antibody aggregation comparable with other analytical techniques that are industry standard, but much quicker. Now, if the focus of our research is to understand the composition of the complexes that we may have in the sample, meaning complex stoichiometries or oligomeric states, we can also use mass photometry. In fact, mass photometry can be used to characterize complexes that are formed by a combination of DNA, RNA, or protein. In this case, the aim was to characterize a compact RNA-guided nucleases. In fact, the nuclease that was to be characterized was Cas12F. And first, they started measuring the nuclease on its own. They started saying that it exists predominantly as a monomer. But as soon as they add a guiding RNA sequence into the mix, they start detecting the complex that is formed by two nucleases and one guiding RNA sequence. And finally, by incorporating into the mix the target DNA sequence, the species that was detected and new or was the catalytically active was the one corresponding to two nucleases bound to a guiding RNA molecule and acting on a double-stranded DNA target. So these type of assays where the focus is on characterizing stoichiometries of complexes formed by different types of biomolecules can quickly be done using mass photometry. Mass photometry can also facilitate the study of membrane proteins that sometimes, because of their nature and the requirements to remain soluble in aqueous solutions, may be challenging to characterize in other systems. This data was generated by Philip Kitchen, who's going to be our next speaker, and Kaolan Brown at the Aston University. Here it's shown how aquaporin 4, which is an integral membrane protein, that is commonly found on the central nervous system and is a promising drug target. In this application, we can see that the aim was to monitor the sample abundance of the M1 isoform of aquaporin 4 that was purified and embed in small nanodiscs as a part of a functional and structural characterization process. The histogram on the left shows how aquaporin 4 tetramers embed in a small that will have a mass of roughly 310 kilodalton are present on different fractions of a purification process. So clearly in fraction 3 is where we have the highest abundance of the complex that they were aiming to characterize. And of course, there were higher order oligomeric species that were also embedded in nanodiscs. So in order to confirm that this was the aquaporin 4 tetramer, researchers did an assay mixing with a positive control antibody or an antibody that was able to detect the aquaporin 4, and also an antibody that acted as a negative control, an anti-GFP antibody. So here in gray, we see the negative control, and we see mostly free antibody and the aquaporin aquaporin 4 complex. But on the positive control, we not only see a little bit of the free antibody, the aquaporin complex, we also see a species at around 449 kilodalton, which corresponds to one antibody binding to the aquaporin 4 complex. So overall, mass photometry was used to examine the composition and quality of the protein samples at different points during the purification process. And also mass photometry helped to identify the sample components and characterize isolated nanodiscs embedded in aquaporin 4 singlets prior to further study. Finally, we can gain insights on how mass photometry can help us to understand how different species interact with each other under different conditions or depending on different components. The example that I am going to be showing on this slide shows how mass photometry can be used to understand how biomolecules interact under different conditions. In this case, Herceptin, which is an antibody, a monoclonal antibody that is used to treat cancer, breast and stomach, I think. This antibody binds to the HER2 receptor, which is its antigen. And what we can see here is that if the antibody Herceptin and the HER2 antigen are mixed at a 1-to-1 proportion, we obtain mainly the 1-to-1 interacting species. However, if we were to mix an excess of the antigen along with the antibody, what we detect is not only the 1-to-1, which is in yellow, what we detect is not only the 1-to-1 interacting species, also the 1-to-2 interaction species. So this highlights how mass photometry quantifies the occupancy of a binding site of antibodies at different conditions. And these assays take around less than 1 minute in terms of measurements. Overall, mass photometry provides single molecule detail of samples measured in the bulk, so it really combines the best of both worlds with regards to single molecules and ensemble techniques. This enables the extraction of information of subpopulations, such as oligomeric states and complex stoichiometries. Additionally, because mass photometry is a single counting technique, and it does it over a wide mass range and high dynamic range, it provides information on sample homogeneity and the detection of low abundance species. Therefore, mass photometry can be employed as a screening tool that streamlines the analysis workflows and saves times on dedicated instruments that could potentially be more time or sample consuming. The data that I'm showing here corresponds to the specifications of the 2MP, which is our second generation mass photometer that we have available at Refeyn. And in fact, this is our most versatile instrument. The 2MP can detect particles between 30 kilodalton and 5 megadalton and operates at a concentration range between 100 picomolar up to 100 nanomolar. It has a mass precision of 2%. And overall, it can tell apart the species, a general rule of thumb that have a mass difference of 10%. At Refeyn, we not only developed mass photometers, we also made available a whole line of consumables to facilitate mass photometry measurements. Overall, the aim is to make available to our customers hassle-free measurements with reduced samples of preparation for them to have greater confidence from consistent mass measurements in mass photometry. Mass photometry has been in the market for a little bit over 5 years, and since then, we have been very well received by the scientific community, both in academia and industry. In fact, we have been able to position or to install 300 systems worldwide, which have enabled the characterizations of molecules in a way that was not possible before. Although we are headquartered in Oxford, we also have a strong presence on the American market, and we are expanding in Asia as well. At Refeyn, we are pioneers in analytical instruments that put mass photometry technology within the easy reach of scientists, as we believe that this technology can help accelerate science by making sample characterization more straightforward and make it possible to analyze complex assemblies and interactions in a new way that was not possible before. With this, I will finish my presentation and I will hand over to the moderator. Thank you.
Thank you, Perla. At this point, I'd like to introduce our next speaker. Philip Kitchen is a BBSRC Discovery Fellow at Aston University. His research interests are in the structure, function, and physiology of mammalian membrane proteins, with a focus on water channels, also known as aquaporins, ion channels, and G protein coupled receptors. Philip, please take it away.
Hello, everyone, and welcome. My name is Dr. Philip Kitchen, and I'm a BBSRC Discovery Fellow based at Aston University. And I'm going to be sharing with you today some of the experiments in which we've been using mass photometry. So my lab's primarily interested in membrane proteins, which come in all different shapes and sizes. The ones that we're particularly interested in are these ones here and here, integral membrane proteins, which are inserted into the lipid bilayer. And primarily we're interested in channel proteins, so for ions and water, transporters, usually ATP directly dependent transporters, and G protein coupled receptors. Why we're interested in membrane proteins? So many of you might have heard this statistic that whilst membrane proteins make up quite a small fraction, about 25% of the proteome, they account for over 60% of drug targets. And so understanding the structure and function of membrane proteins is a great way to develop new drugs, and that's primarily what we're trying to do. Okay, so in order to make proteins for study, this is our typical setup. We express our proteins in Pichia pastoris. We use this primarily because it's a eukaryotic expression system. Many mammalian membrane proteins won't fold properly in a prokaryotic system. And Pichia also has lots of the enzymes required for the post-translational modifications that are often quite important for membrane protein functions, such as glycosylation, palmitylation, and phosphorylation, and so on. Typically grow them in this kind of fermenter setup that's shown on the slide, and then we'll isolate our plasma membranes and solubilize them using either the traditional approach of detergents or using a more novel approach, which is nanodisc forming polymers such as styrene maleic acid. So why the two approaches? Especially for mass photometry, I think this difference is really important because when you solubilize a membrane protein using detergent, you always have to have detergent present above the critical micelle concentration of the detergent. Otherwise, the micelles fall apart, the detergent falls off your protein. Because it's hydrophobic, it's going to come out of solution, you lose your sample. So all downstream work done with detergent solubilized membrane proteins has to have detergent present above the critical micelle concentration, which typically means that your sample is going to contain lots of empty micelles. This is bad for mass photometry because the mass photometer is going to see your empty micelles, and there's typically going to be quite a lot more of them than the micelles containing your protein. And so it's very difficult to make a measurement. So our alternative approach is to use styrene maleic acid or related polymers. And what this does is acts kind of like a membrane cookie cutter, punches out discs of lipid from the membrane being solubilized. Some of those will contain your protein. And the really great thing about this is that once the polymer has formed the nanodiscs, you don't need to add free polymer. The polymers are stable, so you can do your solubilization, wash away the free polymer, and then you get a sample containing only your nanodiscs. And so this is much cleaner for mass photometry work. So with our SMA solubilized proteins, we can do all of the same purification techniques that we would typically use for detergent solubilized proteins. And for us, this is usually a two-step purification using immobilized metal affinity chromatography with his-tagged proteins, and then size exclusion chromatography for a second cleanup. And from the size exclusion trace, you can see that we're able to purify our proteins solubilized in SMA. And what you can see on this trace is one of those key differences between SMA and detergent. So the detergent solubilized protein in red elutes later, so it's smaller. If we look at this peak here, than the protein solubilized in SMA. And that's because the SMA solubilized protein comes along with that disk of lipids and encircling polymer. So we can already see that in the size exclusion traces. So we're nicely able to separate out our protein from any purification contaminants. And when we run the protein on Kamasi gel, it looks very similar to what we get in the detergent. But already here in this gel on the right, you can start to see one of the benefits of the polymers compared to detergents. So in this DDM, detergent solubilized sample, you can see there's an additional band down here that's starting to appear. This protein in particular has some SDS resistant structure. And so the properly folded protein runs at a slightly different molecular weight on the gel to denatured protein. And so this DDM solubilized protein is already starting to degrade. And then this is just a more comprehensive look at that. So this is a detergent and an SMA solubilized protein that we've then left in the fridge or out on the bench for up to 2 weeks. And hopefully what you can see is the band in red is the degraded protein, the band in green is our intact protein that we want. When we solubilize in detergent in the fridge, it's completely degraded by 3 days, and out on the bench, you've got no chance it's gone in a day. Whereas when we solubilize with SMA, it's stable in the fridge for up to 2 weeks, and even overnight on the bench, it's fine, although it started to degrade by 3 days. And so this really can help to simplify membrane protein workflows, because with a detergent solubilized protein, you really need to purify it and then do your experiments immediately. With SMA, you've got a bit more wiggle room, up to 2 weeks for this protein. And then on the right is just another way of showing something similar. So this is a protein unfolding experiment where we use a cysteine reactive dye and ramp up the temperature. And as the protein unfolds, exposed cysteines become accessible, the dye binds, and we see a fluorescence signal. And you can see there's a clear shift in the unfolding temperature between the detergent solubilized protein in red and the two polymer solubilized proteins in blue and green. And you can do all of the same downstream analysis with SMA solubilized proteins that you would do with detergent solubilized proteins. So here are just a few quick examples where we've used tryptophan fluorescence quenching to quantify binding of a small molecule to our protein. And on the right are some 2D classes from an ongoing cryo-EM project of the SMA solubilized protein. And so here's a mass photometry trace of 3 of the proteins that we work on. And you can see it works really nicely. We get nice, fairly homogeneous peaks. And what you can also see here in green, it's a protein that I've just left out on the bench for a bit to degrade and aggregate. And you can really clearly differentiate between the correctly folded protein in yellow here and the aggregated protein in green. So this provides a really nice and quick way to assess sample quality. Okay, so what I thought I would do is just go through 4 quick examples of experiments that we've got running in the lab at the moment where we're using mass photometry. Okay, so the first one of those is we're looking at solubilizing a G protein coupled receptor and trying to optimize the solubilization so we get the best functional protein. We're characterizing the protein function using tryptophan fluorescence quenching. So on the left is some example spectra of unbound protein in blue and ligand-bound protein in purple. And you can see is there's a right shift of the tryptophan fluorescence spectrum, which we can then quantify as a ratio between the intensity at 2 points on either side of that peak. And so that's what we've done on the right-hand side. We solubilized our protein with 2 different polymers, and then we've looked at ligand binding by tryptophan fluorescence. And there's 2 interesting things that we can see here. The first is that there's about a 10-fold shift in the apparent binding affinity between these 2 polymers. And the second is that the bottom of the black curve is much, much lower than the bottom of the blue curve, so that when the ligand does bind, it looks like the protein's ending up in a very different conformation in the two polymers. So then we wanted to try and compare the binding affinities to the particle sizes, because one of the things that we thought was that maybe the different polymers are given as different particle sizes, leading to different lateral pressures in that disk of lipids that comes in the styrene maleic acid lipid particle. And that if that lateral pressure is too high, then the protein won't properly be able to undergo the conformational changes that it needs to for ligand binding. And if that lateral pressure is too small, then the protein will be kind of flopping about a bit and exploring conformations that it wouldn't be able to in a native membrane. And so what was interesting was that we found by mass photometry that the major peak that we get is different by about 30 kilodaltons between the two polymers. And so to try to explore this a little bit more, we use size exclusion chromatography to separate out different sized particles from the better of the two polymers from the previous slide. And again, what we saw here was not quite such a pronounced difference, but still a difference. So again, a shift in the binding affinity and a difference in the bottom of the curve. And so if we put all of that together and look at how particle size might affect the binding affinity, it looks like what we might have is a nice sweet spot of particle size where the protein is exploring the right conformations, but it's not held so tightly that it can't properly bind to the ligand. And if you go too far either side of that optimal size, then you start to lose binding affinity. So obviously this is only 3 data points. So what we're planning to do next is a size exclusion chromatography experiment where we collect very small fractions and characterize each of the fractions one by one for the binding affinity to try and plot more comprehensive curve of the binding affinity against the particle size. But there's really interesting preliminary data showing that possibly the particle size is really important for getting functional protein. And that's something that we can characterize by mass photometry. Okay, so this is a second example now. This is the protein that my fellowship is based around. It's aquaporin 4. It's a water channel protein, and it's expressed very highly in the brain, and specifically in astrocytes. I won't go too much into the biology here, but what's really important on this slide is that if you look at this electron micrograph on the right-hand side, so this is a freeze fracture electron micrograph of astrocyte membranes. And what you can see are these large square arrays of protein. And it turns out that that's aquaporin 4. And what happens is that aquaporin 4 tetramers, so the native structure is a tetramer, pack against each other, and you get these large oligomers of tetramers in the astrocyte membrane. And a big open question in the field is, what are these for? And they're also clinically quite important because it turns out that in an autoimmune disease called neuromyelitis optica, these large arrays, orthogonal arrays of particles, are the autoantigen for an autoantibody that causes neuromyelitis optica, demyelinating disease. And so we want to understand a little bit more about how these orthogonal arrays of particles assemble in the membrane. Okay. And we were inspired by a recent study that used single molecule turf microscopy to try to characterize the kinetics and thermodynamics of this assembly process. And these guys developed this model for the initial stages of OAP assembly, where you have an equilibrium between monomeric tetramers, dimers of tetramers, and trimers of tetramers. And so we thought that maybe if we can solubilize aquaporin 4 containing membranes with styrene maleic acid, then we can trap those oligomers in their state, because once they're extracted, if you've got three tetramers in a disc, then they're stuck together. If you've got two in a disc, they're stuck together. And so we can get kind of a snapshot of the oligomeric state of aquaporin 4 in the host membrane. Okay, so this is what those samples look like on the mass photometer. And we see exactly what we were expecting. So a peak that we think corresponds to a single tetramer in a lipid nanodisk. So the size that we're seeing there is the size of the protein tetramer plus the lipids plus the polymer. And then we get the multiples of that, which we think correspond to 2 tetramers in an SMA disk and 3 tetramers in an SMA disk. And if we do the solubilization under different conditions, we can then quantify the ratio between these different populations. And this is helping us to start to get a handle on how the membrane environment can facilitate this assembly of these orthogonal arrays of particles. Okay, so here's another example from a new project that we've very recently started working on. And when you first start working on a new protein, you've expressed it, you've purified it, you want to check that what you've purified is the right thing, it is what you think it is. And so one of the ways you would often do that is by Western blotting against either the protein that you've expressed, if commercial antibodies are available, or against an epitope tag that you've put onto the protein. And so on the left-hand side is just an example of a Western blot of this protein. It's a heavily glycosylated protein, so we see a very large smear at high molecular weight. And so one of the things that we've started to do now is, because Western blots are a long and laborious experiment, typically 2 days, and mass photometry is a very quick experiment, maybe 2 minutes, we've been exploring whether we can replace Western blots to validate our samples with mass photometry. And so the basic idea is that you take your protein, you put it on the mass photometer, you then add an antibody against your protein, put it on the mass photometer again, and look for a mass shift that's consistent with antibody binding. And so that's an example of exactly that on the right-hand side. In green is the free antibody at roughly 150 kilodaltons, that we'd expect to see it. In blue is our purified protein. And then in yellow is the two together. And you can see that there's a new population that appears when we mix the antibody with the protein that's shifted by about 150 kilodaltons, consistent with a single antibody binding to our protein-containing SMA particle. And at the same time, we also see a reduction in the amount of free antibody, which again is consistent with antibody binding. And what's great about doing this is that what you also get is some information about the oligomeric state of the native protein. So in a Western blot, you would typically run an SDS page gel, which is going to destroy the quaternary structure of your protein. Whereas on the mass photometer, we're running our native proteins. And so by comparing the size that we see on SDS page gel or Western blot with the size that we get on the mass photometer, we can start to get a handle on what the native oligomeric state of the protein is. And in this case, we think we've got a protein that's purifying primarily as a trimer. Okay, and here's, this is the last one. So this is another new project that we've just started looking at binding of peripheral membrane proteins to, in this case, empty styrene maleic acid lipid particles made from model membranes, liposomes or something similar. And yeah, so the basic idea here is similar to the one on the previous slide, where instead of adding antibody and antigen, we're now adding our lipid discs with our protein that we think should bind to lipids. And on the right is an example of the kind of mass spectrometry trace that we've been getting. So in orange here is the protein alone. In the slightly lighter orange, here is the lipid nanoparticles alone, and in blue is the two mixed together. And so there are two important things here. Firstly, the protein peak disappears when we mix the two together, and there's a rightward mass shift of the lipid nanoparticles, both consistent with the protein binding to the nanoparticles. And so using this, we can start to characterize the lipid binding specificity of this protein by making model membranes from different lipids. Okay, so I thought I'd finish off just by explaining a little bit why we like mass spectrometry so much. So first of all, it's a very simple technique. The underlying physics behind how it works are very complicated, but in terms of using the instrument, it's very much plug and play. We've got students in the lab using the instrument with no problems, so it's really quite easy to use. I think it gives a nice quick assessment of sample quality and heterogeneity. So, for example, for the cryo-EM project that I showed a little bit earlier, one of the routine things that we're doing now is after solubilizing the protein and purifying it, we'll put it onto the mass spectrometer to check, is it a single peak of the right size, before we then go on to do the expensive cryo-EM experiments. Another key benefit is the low sample requirements. So typically you're using a few microliters of a protein at nanomolar concentrations. And so that's great for us because one of the things that we're interested in is comparing how our proteins behave when they're expressed in a microbial system versus how they're behaving when expressed in a mammalian system, kind of a more native system for the proteins that we're interested in. And that can be very challenging, especially for membrane proteins, to get enough protein from a mammalian expression system that you can actually then go and do something useful with it. And so this very low sample requirement has started to make mammalian expression systems a little bit more accessible for us. Okay, I thought I'd just describe a little bit as well about the challenges that we faced. So one major thing is cleanliness. It's a single molecule technique and very sensitive to any kind of small dust particles and so on. So it took us a little while to kind of get used to working at the level of cleanliness required to collect good data. So that's something to keep in mind. And the second thing is that because the technique works best at low concentrations and quite poorly at high concentrations of protein, if you're looking for a binding interaction of low affinity, then that can be very difficult, because if you dilute your complex down to the concentration that you need to look at it by mass spectrometry, then you go below the KD of the interaction and everything falls apart. I understand that there's a new microfluidics-based system to try and get around this a little bit, and I'm sure Perla will be happy to speak about that in the questions if anyone's interested. Okay, so finally, just like to acknowledge all of the people involved in all of the different projects that I've spoken about. The people at Refeyn who've been really helpful for us, both in terms of kind of getting our instrument set up and troubleshooting. And yeah, so thank you for your attention, and I'd be happy to open up the discussion with the audience. Thank you.